Okay, what I wanted to do today was to start off by going through what's going to be on the final, and this is up on Blackboard now. So if you got on the Blackboard and looked at the Unit 4 material since probably 12 yesterday, you'll see that the last two vaccination lectures are up now with the mini lectures. There are two sets, or there are two past exams that are up, not containing any of the cumulative, but containing new information, so the immunology stuff. I also went through there and crossed out questions. You can still see them physically, but just the lettering on it is crossed out, so you can't actually tell, or so that you know that's not a question that I'm asking about. There is some material that we used to go through that we just did not end up covering this semester, so I crossed that out for you. There is a key for one of those as well, and then this is up for each individual class because the objectives from the student presentations differ between this class period and the 1 o'clock class period, just so that you're aware. So I figured I'll take a few minutes to go through this and then answer any questions that you have about the final before we move on for the rest of the day, spending more time with starting with toxins and pathogenicity factors and then moving to some more general questions if we have time. So big ones for the immune system and pathogenicity, there's 6. Describe the process of phagocytosis and its contributions to immunity. In other words, be able to write out the steps of this process. We've talked about everything that's involved in this process from the attachment step through the destruction of the microbe, and then ultimately the presentation of that microbe on MHC2 of macrophages and B cells. So that all encompasses 11.1. 11.2, describe how inflammation contributes to the immune response. Making sure to focus on terms like diapedesis, margination, understanding the process of chemotaxis of those neutrophils to the site of infection are all important concepts to review. Discuss the influence of virulence factors on pathogenicity. That's a conversation we'll continue today, but we have talked about this in part throughout. We've talked about the role of capsules in preventing phagocytosis. We've talked about a couple of examples of where organisms will bind to the Fc region or destroy the Fc region on an antibody as a way to block the opsonization in phagocytosis. And then today we'll talk about toxins. Explain the function of B, T helper, TC. Basically, know what they do, know how they do it. Within that, you should be able to know the different cell types and what they have on them. So for example, we talked quite a bit about the difference between T helper and T cytotoxic. What do they recognize? Which MHC complex do they recognize? And what is the CD co-receptor, CD4, CD8, that they use to recognize MHC1 or MHC2? Compare NK and T cells, or TC cells, similar function, different way to getting to that function though. And then finally, describe how antibodies and T helpers enhance phagocytosis. And we've talked about opsonization and the role, the structure and the function of antibodies within that. These questions are likely to be short answer and essay on the exam. Okay? There may be a couple that are maybe fill in the blank or circle this region of an antibody or something like that. I haven't completely finished these questions, but for the most part they're going to be short answer or essay based. Because most of the multiple choice and fill in the blank are based off of your presentations. Okay? And that part is largely written actually already. I'm not going to go through every one of these because as you will see, because some groups chose to have multiple different objectives. There are 26 in this section. Okay, So, will all 26 be addressed? No, I don't have the space for 26 questions on the exam. I think the basic thing to describe how I'm testing this section is know and describe. Be able to know the basic information. And then be able to describe by giving examples. So for example, we talked about, I'm going to go to Carly, you guys talked about extremophiles, and we said, where would you find a hyperthermophile? Okay, you brought up the example of deep sea vents as being one example. So one potential multiple choice question would be, which of these locations is a place where you might find a hyperthermophile? Okay. So the point that I'm trying to make here is that these questions are not necessarily intended to trick you up. They are basic knowledge type questions. Okay. And that's the thing to keep in mind. The review questions that you have all put together for this section, and your classes is actually quite extensive. Those review questions are what I am basing my questions off of. They will not be verbatim, they may be similar, but taking the time to go through those questions and come up with answers to them will help you on this section of the exam. Questions on that section? And then the last, which we have not gotten to yet, but this will become more apparent in the next couple of class periods. On Wednesday, we'll talk about identifying immunity as being passive or active, natural or artificial. This is really the first two slides of the next mini lecture. Cover this basic concept. All right. Compare and describe categories of vaccines and how they stimulate the adaptive immune response. We'll talk about 6 different types. And for each of those types, what I would like you to know are some of the basic information on each of them. What is the structure of those vaccines, for example? And then what parts of the immune response do they actually stimulate? And the answer really comes down to, are the antigens capable of being presented on MHC1 and or MHC2? And that's where that's where you'll see the mini lecture kind of focuses on that concept. And then the last two become our conversation on Friday. Describe herd immunity and its function, and the function of its design. That is the basis for the mini lecture. And then what we will spend our time in class talking about, why do folks choose not to vaccinate? And I'm not going to ask anybody in here to disclose whether you support or refute it. This is more just a hypothetical. For somebody that chooses not to, or chooses for their children not to, why not? And then how do we strategize to talk to such individuals, which I think is a really important conversation, particularly in this class, given the high proportion of you that are going into medical profession in some way. Okay? So that's where we'll spend the majority of class time on Friday, but keeping, again, this conversation focused or grounded in herd immunity as an example. Below this, you will find what is fair game for the cumulative section. I will be pulling together 25 questions, each worth 2 points. They will be multiple choice, matching, or fill in the blank. And they may cover the following. And I've put may here because it may not be in there, but these are the areas you should be studying. Morphology and gram status or gram status comparisons. For example, what makes a gram positive different from a gram negative? Which one has a thicker cell wall? General themes of metabolism, fermentation, and final electron acceptors. Growth characteristics. Can we define an organism as a mesophile or an acidophile based on a particular growth characteristic? And stages of growth, that lag, log, or exponential, stationary, and death phase. Ways we can control growth. So there are two major classes of toxins, or two major categories, exotoxins and endotoxins. The name in part tells us about, the name maybe is probably actually misleading, I would say. But exotoxins we can typically think of are secreted from an organism, whereas endotoxins are actually part of the organism's structure. Both gram-positive and gram-negative microorganisms can produce exotoxins. Doesn't necessarily mean that they will, but they can produce exotoxins. Endotoxins are also known as lipid A, which is part of the lipopolysaccharide. Where do we find lipopolysaccharides? On what type of organisms? Not gram-positives. It's on the gram-negatives. It's on the outer membrane of the gram-negatives. So the lipid A portion is actually what we classify as the endotoxin. Now, these can trigger a very, very strong immune response, particularly in mice. It actually triggers an incredibly strong immune response. But it takes a lot of it to trigger that immune response. So endotoxin typically we think of as being less virulent, less likely contributing to the disease and the signs of the symptoms of the microorganism. Exotoxin, on the other hand, is oftentimes much stronger. A couple of other differences. Exotoxin is typically what type of macromolecule? Sugar or carbohydrate, protein, lipid, or nucleic acid. Typically protein. And that also indicates to us proteins we know that if we heat them to a certain point, they can denature. So exotoxins are heat sensitive. Beyond a certain temperature, they actually can be denatured. They lose their three-dimensional folding, and therefore they lose their toxic effect. We use this to our benefit. In what ways do we use this to our benefit, to human benefit? This may become more apparent on Wednesday. Who's had a tetanus shot? Okay, majority of class. Who's had a pertussis shot? DTaP is all the same thing. Those are inactivated toxins. We're not actually injecting into you the bacteria, it's just the toxin that's being injected. It is what we call a toxoid vaccine. So toxoid vaccines contain the heat-inactivated form of the toxin with the goal in mind of producing antibodies that would neutralize or opsonize these toxins to inactivate them, to prevent them from having an effect. A couple other features I just want to point out real quick. Endotoxins cannot be converted to a toxoid vaccine because they are incredibly heat stable, unlike the exotoxins. Let's see, it lists this as part of the cell wall, but it's more the outer membrane. And then because endotoxins are part of that LPS, we only find them in gram-negatives, unlike the exotoxins, which can be found in both gram-negatives and gram-positives. Now, there are multiple different types of exotoxins. There are some that help to lyse cells, which we would call cytolysins. If they lyse red blood cells, we call them hemolysins. There are some that have neurotoxic effects. Botulism and tetanus both have neurological effects, so we call these neurotoxins. There are others that have effects on the epithelium lining the gastrointestinal tract and lead to diarrhea, because what's happening is it's forcing the calcium out of the cells, depolarization of calcium, also leading then to secretion of water from cells, which leads to the watery diarrhea that's associated with it. So there are a number of different types, and we're not going to spend the time talking about the function of these different types per se. Probably the only one that, I would say the only one that we're really going to talk about are these cytotoxins, the hemolysin, and then a leukocidin is just one that would particularly particularly lyse a white blood cell. Yeah, I think that's all I wanted to mention there. In the case of the endotoxins, where we find these being released is a couple of scenarios. One, if we get lysis of those gram-negative cells, maybe by an antibiotic leading to the lysis, for example, we can find release of endotoxins. The other case is here, in which we have a pathogen being broken down within a macrophage. The destruction of that might end up secreting or releasing the endotoxin from the organism, leading to secretion of cytokines, leading to an inflammation response. And so occasionally, if an individual has bacteremia, bacteria within the bloodstream, and those bacterial cells are lysed, what we can find then is that the lipid A can trigger quite a strong immune response, leading to effects like shock. Because what do cytokines do? What is the role of a cytokine in terms of inflammation? So what effects do cytokines have in inflammation? Vasodilation is one of the biggest ones, and also the edema that's associated with it. But particularly that dilation is what's going to lead to decrease in blood pressure and ultimately shock in the individual. I wanted to show this, wanted to show this picture real quick. So I think we alluded a little bit to the fact that some of these organisms have hemolysins, and we alluded to what's known as a blood auger plate. So this is a plate where it's been spiked with sheep's blood basically. And this is an example here of Streptococcus pyogenes, but we would see the same effect with Streptococcus pneumoniae as well, and that pneumolysin that it produces, which is an example of a hemolysin. The pneumolysin would be secreted from cells that are being, and remember what's different about pneumolysin is Strep pneumoniae is actually lysing. It's undergoing an autolysis to release that pneumolysin. And as the pneumolysin would diffuse into the media, it lyses those red blood cells and leaves behind this halo effect that we observe here. What is the benefit to the organism? So what does lysis of red blood cells release? High concentrations of iron. And we commented on that iron is a limiting resource for most microorganisms. Most organisms have mechanisms to scavenge up excess iron and capture it on the surface of the cell and then import it into the cell. These iron scavenging molecules are known as siderophores. And so in the case of Strep pneumoniae, Strep pyogenes, other organisms that have hemolysin activity, what we will find is they'll capture that excess iron that might be found within those red blood cells, bind to those siderophores, and then import the iron to support their own growth. So this is how we can find the hemolysin and siderophores being used at the same time. Who had Pseudomonas fluorescens as their unknown? Somebody in here. Derek, you guys did, that's right. So did we look at the fluorescent color on it? So the fluorescence is actually the siderophore that Pseudomonas fluorescens produces. So at high concentrations, iron is limiting. It turns on the expression of that siderophore, which is actually fluorescent when you hold it under UV light. Okay. I'm just going to bring this one up real quick too because I did comment on it. Superantigens are not classified as an exo or endotoxin necessarily, but can be an exotoxin. Superantigens are molecules that actually stabilize the interaction between the T cell receptor and MHC2. It does so. What did we say about T cell receptor? Does it recognize a specific epitope or antigen, or does it recognize a general pattern? It is specific. So B cell receptor and T cell receptors both recognize a specific antigen that's loaded in the MHC2 complex. Superantigens here will actually stabilize this interaction between the T cell receptor and MHC2, or particularly the antigen in MHC2, even if there's not a match. Even if this T cell receptor is not designed to recognize that antigen. And so what this leads to is an activation, an abnormal activation of T cells. Typically within your body, you've got, or in response to an infection, maybe 0.001% of T cells are activated. In the presence of superantigens, this could lead to the activation of up to 20% of your T cells. It's like 2 million times, 200,000 to 2 million times more than what we would normally find. What do T helper cells secrete? Cytokines. And we can already start to see where that's going to activate B cells and T cytotoxic, but also some of those cytokines are going to activate macrophages, neutrophils. We're also going to find responses of inflammation, vasodilation, edema. And those effects right there, what we find oftentimes with these superantigens is, again, what we call toxic shock syndrome. Individual goes into shock because of the mass efflux of cytokines from these activated T cells. One of the areas where this is actually of concern right now, nobody knows the reason why, but since about 2011, the number of cases of scarlet fever in the US have gone up by about, an order of like tenfold. So if you compare the previous 10 years before, from like 2000 to 2010, it was only a couple of cases, or maybe like 20 cases per 100,000 individuals. Now we're up to like 200, 300 cases per 100,000 individuals since 2011 through now. And nobody knows why. It doesn't necessarily seem, it doesn't fit the trend of antibiotic resistance. That would be more gradual. But the rash that is associated with scarlet fever is actually due to superantigens binding T cells to a macrophage indiscriminately, and then leading to responses of inflammation, redness, fever, swelling, all classic signs of inflammation that are associated with scarlet fever. Okay. All right, any questions on toxins, particularly the contrast between exotoxins and endotoxins, or superantigens? Deb?
You say that like inflammation and edema can be triggered. How is that triggered again? Is it from cytokines?
Yeah, so Devin's question is how is the inflammation actually triggered? How do we get vasodilation and edema? And the short answer, Devin, is yeah, it's from the cytokines that are doing this. One of the things that we did not talk about is cytokines not only have an effect on recruiting neutrophils through chemokines, but also some of those cytokines will actually bind to the endothelial cells that line the blood vessel, and it causes those endothelial cells to begin to change shape, which leads to the vasodilation, leads to some of the spacing between them for the edema to occur, and also creates the spacing for the diapedesis of those neutrophils in between those. We'll just kind of forego them. But what I wanted to do was to just touch on a couple of effects real quick. So does anybody not have the worksheet? Okay, so I will hand out the worksheet to those who need it from Friday. But in the meantime, what I wanted us to work on was, let's take 5 minutes. And let's go ahead and work on number 9 and 10. And if you want to, you can move on to 11 as well. Yeah, I do. exo and endotoxins, but in your own words. So what are some of the features of exotoxins and endotoxins that differ from one another?
Endotoxins are lipid and exotoxins are protein.
Okay, endotoxins are lipid, exotoxins are protein. What else? Derek.
Endotoxins are less virulent, while exotoxins are stronger.
Good. And perfect. So exotoxins takes a lot less to trigger an immune response. Okay. And when I say this, I suppose I need to clarify a little bit that the exotoxins will trigger a very strong immune response, in that it takes very little for there to be T cells and B cells, et cetera, that are activated in response to it. I think where I may have been misleading in the mini lecture for those of you that listened to it, as I said, the endotoxins do trigger a strong immune response. I think what I meant to say was it triggers a really strong inflammation response when it's being produced. Not all exotoxins necessarily trigger a huge inflammation response, in part because of where it's localized. So for example, some of those gastrointestinal ones, that's kind of a region where it may not necessarily trigger as strong of an immune response. But what we find in humans is it takes a lot of endotoxin to cause disease on the level of milligrams per kilogram versus some of these exotoxins. We're talking nanograms or 1/10 of a nanogram needed per kilogram. I think I calculated last year in micro that you need to grow about 100 milliliters of Clostridium botulinum and then isolate the toxin to kill me. I think that was what we figured it out as. A bit morbid, but some people probably were about ready to take me up on the offer. What else? Other differences. Which can be used as a vaccine? Exotoxins. Which one is more heat stable? Endotoxins. Which one is secreted? Exotoxins. Good. Which one are only found in gram-positive or gram-negative organisms? Good. Endotoxins only in gram-negative, which kind of helps us to answer number 10 here. So if your friend knows that an exotoxin is produced in Strep pneumoniae, how do you know that your friend is correct? What are the characteristics of Strep pneumoniae? That's a gram-positive organism, yeah. Okay. And then just as a reminder, if something is considered, 1, secreted, and 2, can lyse a red blood cell, what does that tell us about the toxin? Since it's a secreted protein, that tells us it's an exotoxin. What specific type? Hemolysin in this case, exactly. Okay. The things that we did not cover for time reasons included number 12, which talks about coagulase and streptokinase. Number 13 is there probably for you to use as a review question. Okay, but 12 won't be covered on the upcoming exam. All right, so what I want to do now is hand out a few. I think that this is a great one because it just helps to remind us what are the specific features of these different cells that we find in the immune response. So the question is, which immune cell or cells are considered or have these general characteristics? So for example, which ones are considered professional antigen presenting cells? Or in the second one, which ones are involved in the process of phagocytosis? Why don't we take, let's take for the sake of time, let's take 2 minutes and let's just go through A through D here. I want you to try with A through D your best to come up with answers to those. If you get done with those ones in 2 minutes, keep going. But I'll keep time and then we'll discuss those ones afterwards. How many of you feel confident to do this without your notes? Couple. Okay. This is a good thing. This question, I think, maybe out of any other, is probably the best study tool for immunology because it connects so many topics that we've talked about. And this is where the nuts and bolts, the basic information is, and helps you to start to mentally organize some of the functions of these various cells. Okay? So let's take a couple of minutes to go through a few of these, and we'll see where we end up. So which of these cells, B, T helper, T cytotoxic, neutrophil, macrophage, and NK cells, let's start with B actually. Which of these are involved in phagocytosis? And I'll give you a hint, there's 3 of them. Abby? Yeah. So our 3 phagocytes are B cells, macrophages, and neutrophils. The follow-up question here is, what's the definition of a professional antigen presenting cell? What do professional antigen presenting cells have to be capable of doing? Derek? Not detecting, but presenting something on MHC2. And in other words, what else has to be true for them? If they're presenting on MHC2, what do they have to have the ability to do? How do we get presentation on MHC2? What process leads to that? Yeah, it is the phagocytosis. In other words, our professional antigen presenting cells can only come from the subset of cells that are involved in phagocytosis. So of these, there are 2 of them that are considered to be professional antigen presenting cells. As Derek pointed out, they have to present MHC2 on the surface of those cells. Which 2 are they? Yeah, our macrophages and our B cells are our professional antigen presenting cells. Which cells recognize MHC1? TC. Any others? Or the presence, right? If it's present, there's an inhibitory signal that prevents them from being active. So in this case, actually, you could consider both TC and NK cells to be ones that recognize MHC1, but their response to that recognition is different. Which ones are part of the adaptive immune response? Three of them up here should be part of the adaptive immune response. Yeah. B, part of the adaptive, TH, oops, sorry, and TC are 3 adaptive. Okay. Let's go through a couple of more of these, and we'll save these review, the additional 3 questions on the back. We'll reserve that for review on Wednesday evening. Okay, we'll kind of save those for practice. Which of these recognize PAMPs? Pathogen-associated molecular patterns. macrophages and neutrophils. What are these PAMPs, or what is an example of a PAMP? Anybody recall what a PAMP is? What's that? Lipopolysaccharide would be an example. Cell wall, peptidoglycan. What was that, Brendan?
I said the same thing you just said.
Okay, peptidoglycan could be an example. Teichoic acid could be an example. These are general features that we find on multiple pathogens. So these are common features among pathogens, and these are bound by by our cells that are involved in phagocytosis, with the exception that B cells are not involved here, because B cells bind by a different mechanism. So macrophages and neutrophils have on their surface PRRs, or the pattern recognition receptors, which allow them to recognize PAMPs. B cells, on the other hand, don't recognize patterns. They recognize a specific epitope. And again, that is a characteristic of all the cells that are involved in the adaptive immune response. They recognize very specific antigens or epitopes. Which of these expresses MHC2? In other words, which ones are professional antigen-presenting cells? Macrophages and B cells. Those two are the only ones that contain or that express MHC2. And they are the only ones then that can present that antigen to what? What recognizes MHC2? What cell type recognizes MHC2? Good, our T helper cells. Perfect. Okay, let's do one more. Which cells secrete granzymes at birth? TC and NK cells. Perfect. All right, we'll stop there for today because we've run out of time. Continue to work through this, okay? And take a stab at those 3 questions on the back as well, because those 3 questions on the back are great examples of a potential essay question. All right? Have a great rest of the day.